Good afternoon, and thank you all for coming to this presentation on DNA. It's going to be an interesting talk. Prosecutor Krastek and I will be going back and forth between the science and how the case evolved, and then what the DNA in the case looked like, and then going on to look at the admissibility hearing and so on. So there's a lot of stuff that's going to be happening. What I'd like to start now for a quick talk, maybe 20 minutes or so, is to take a look at what is DNA, what is its biology, and what do we mean by DNA information. Since, as you'll see in this case, there were several match statistics, it's going to be important to get some intuitive understanding, not any formulas or anything, but some good intuition as to what is a DNA match statistic. What does it mean when someone tells you it's 1,000,000,000 to 1? What does it mean? Okay, so let's get started. And we'll begin with DNA. Most of the cells, 1,000,000,000,000 cells in our body or so, have a DNA. This is a picture of a cell, and that's the nucleus. Oh, I should also add that all the slides that you're seeing will be on the Cybergenetics website. So inside the nucleus, there are chromosomes, and we have 23 pairs of chromosomes. And DNA is sort of like a library. It's the instruction code over 3 billion DNA letters that programs bodies on how to develop from one cell into an organism, how to operate, and there's a lot that's in the DNA. If you go down and look at one of the chromosomes, that's the blow-up of a chromosome, you can take a look at one location. A location on a chromosome is called a locus. And for forensic identification, the loci that we care about aren't so much the genes that build us and code for eye color and so on, but they're genes that have a lot of variation in them, sometimes called junk DNA. No one really knows what these things do, but we know that there are many different possibilities. Instead of having maybe 2 eye colors, they can have hundreds of different values. So the system that's used in forensics are called short tandem repeats, and this is how they work. So you have 2 copies, one from one parent, say from the mother, one from your father, and this is a zoom in on one small location. This may only be a few hundred DNA letters. You have ACGT, right? That's it. It's very small. And each of these units is typically a repeating unit that's 4 letters long, like TCCG. And it just repeats. TCCG, TCCG, a certain number of times. So the top one might repeat 8 times. That's what you would have inherited from, say, your mother. So it goes TCCG, 8 times 4, there are 32 DNA letters here they're repeating. And the allele, the particular DNA that you inherit at a locus is called an allele. It's the value of what's ever in that page on your DNA book. The allele from your father maybe has 7 repeats. So TCCG, TCCG, going out 7 times for a total of 28. And there's stuff on the sides. And those numbers of how many, of what the lengths are, that 7 and the 8, is your genotype. If you take DNA from almost any cell in your body, not a red blood cell, but almost anything else, it will have a nucleus. And as I'll show in a second, the laboratory will be able to figure out what your genotype is, what the genetic type of the 2 lengths are of those tandem repeats in the DNA that you inherited from your mother and your father at a particular genetic locus. As you may know, in forensics in the U.S., there are 13 genetic loci that are used, and we'll come back to that in about another hour or so. But right now we're going to focus more on what happens at one locus so you can see what it means and what it does. Okay, so if we want to ask, how do I identify somebody with DNA, what do? We begin with some biological evidence. Say there's some blood at a crime scene. There's a lot that happens that I won't go into in the laboratory. I'll briefly describe it, but I'm not going to spend 10 minutes showing you all the fancy equipment and the pictures and all that. In the laboratory, what happens is the DNA is swabbed, it's extracted, it's purified, and you end up with a certain amount, it's usually a small amount, maybe a nanogram of DNA, which is, I guess it's a billionth of a gram. I guess it's a very small amount of DNA. And that DNA may have a few hundred copies in it, and that's not enough to see. That's not enough to do anything with. So the two main steps that happen in the crime laboratory is first it goes into a machine that amplifies it. That's called PCR. And from the 100 copies you start with, the amplifying process takes about 2 hours, will make millions or billions of copies, enough to be detected by instruments. It goes through, it keeps doubling the number every cycle until it's exhausted of all the reagents that are there. And you end up with just a vast number of copies of DNA that are suddenly not a nanogram, but just billions of copies, all with a fluorescent label on them. And so now it can be detected. And now it goes through a detecting machine called a DNA sequencer that separates these different labeled fragments by their size and detects them. And because it's based on size, you get a signal. We'll look at actual DNA signals later, but the signal might look like a peak of where there's a lot of fluorescence around where that length of 10 happened, and then another peak with a lot of fluorescence where the 10 or the 12 repeats occurred. And so if you have a very simple DNA genotype from an individual and they inherit a 10 repeat from one parent, 12 repeat from the other parent, then you get a signal that looks like a big peak at 10, big peak at 12. But the point is, with simple biological evidence, if there's one person who contributed, you can look at that one location, that one locus on a chromosome, amplify it, detect it, and by looking at the size, get a signal where the first peak will tell you the length of 10, the second peak occurs where a 12 would be, and just read that off. In particular, you're seeing there isn't a 9, there isn't a 13, there isn't a 14. You're just seeing the presence of those two alleles. Okay? Comfortable? So then what happens? So that's the part you usually read about in the paper or on the TV shows, right? They do all this lab work. But that's not what's the most interesting part of what happened in this case. What's most interesting is the information that was extracted from data. So now we're going to talk about how you make an inference and figure out a genotype. We're going to start with a very simple case. Suppose the data are easy. Suppose there are just two peaks. You think there's only one person in the DNA, and you have a genotype of a 10-12. Okay, so you write that down. That's done at 13 loci, each of the FBI's CODIS loci, and which we'll see later. And those pair of numbers, they could be the same. Your mother and father might both have a 10, for example, and so you have a 10-10 as opposed to a 10-12. But those 13 pairs of numbers are written down, and that's the genotype. At this locus, that's the genotype. At this point, there's no suspect. You haven't looked at a suspect. All you've done is you've inferred a genotype from the data. Okay. Now, you may get a suspect. You may get one suspect. You may get 5 people to eliminate. You may get an entire database of the national database with 5 million DNA genotypes on them to make a comparison. But we haven't yet looked at anyone, and now a comparison is made. So in this mocked-up example, the suspect genotype could be from an actual suspect, someone you're eliminating, or one of millions of people who are all being compared at the same time. A comparison is made, and you see that the 10-12 of what was extracted from the biological evidence is the same as the 10-12, that same allele pair, the same genotype value of a suspect. Okay, so that's good. Now you've made some identification. So let me introduce a concept, because you need, if you're going to understand a match statistic, you need to understand probability. Not mathematical probability, but normal probability. Like, what's the chance it's going to rain tomorrow? You know, the odds of the Steelers winning, which is on one end of the spectrum. The odds of some other team winning, I won't mention, which is on the other end of the spectrum. So probability just means what's the chance of something happening based on all the information that you have. How we're computing probabilities, math, we're not worrying about that today. Just the concept that in this case, if you have absolute certainty that this is a 10-12 genotype from the evidence, and there's a 10-12 from the suspect, then you've made an identification, and the probability of that identification is 1. The suspect matches the evidence. You know that. But is it relevant? What if everybody has a 10-12 at that particular locus? Then it might be you've identified someone that, for example, you may say someone has a nose. So just go and pick up anybody who has a nose. But a lot of people have noses. So it's not going to be very discriminating. For that, we need to ask, could it have been a coincidence? This is the alternative hypothesis. Well, maybe it is the suspect, but matching the evidence, but maybe it's the suspect just matching someone random. So if you take a biological population and you do a bunch of lab experiments, typically on a few hundred individuals, you can see how often these alleles from this one locus appear in the population. Some may be very common, others may be very rare. Others, like 9 or 10, may not even appear at all. This gives you a sense of how frequent they are, how unexpected, what the surprise would be to find a particular allele. You can then figure out that, for example, the 10-12 of the suspect, looking at all this data, maybe occurs with 5% chance in the population. Right? If you take all these numbers together and create all pairs, maybe 100 possible values, 10-10, 10-11, 10-12, 13-15, all the different pairs of what alleles you could have inherited from a mother and a father. Typically about 100 pairs of values in forensic DNA. And if you combine the probabilities, which are roughly proportional to the frequency in the population, so if 10 and 12 are not that uncommon, then perhaps seeing them together would give you a probability of 1 in 20, of 5%. So this is going to be used as a denominator in a second. The question is, how surprised am I? If the answer is I see this in 90%, then I'm not going to get much of a match statistic out of it. If I see it in 1 in 1,000 people, I'll get a huge match statistic out of it. So the question is, what's the baseline? How relevant is that identification? And there's a well-established rule, which will be discussed later, called the product rule, that says that if you want to know what the probability of finding a particular genotype, its rarity, from the population, the way you combine alleles is by multiplying the individual probabilities together. You multiply the probability of the first allele times the probability of the second allele. And then if it turns out that the two alleles are different, you multiply by 2, because it could have come from the mother and the father, or from the father and the mother. There are 2 ways of ordering it. So that's where the 2 comes in. And this is just a generally accepted way of looking at how to get a genotype probability if you know the allele probabilities. So let's continue in this example and suppose 10-12 has a relatively typical appearance, maybe 5% of the population. Most allele pairs are in a range of maybe 1% to 15%. So 5% is about typical. Now we make a comparison. And we can say, what's the extent of coincidence? So from the biological population, if it were a coincidence from our frequency data, we can compare the population genotype of all possible pairings, 10-10, 10-12, and so on, and say, well, that would occur with 5% chance. If you pick somebody in the room completely at random, they'd have a 5% chance of having a 10-12. That's just by coincidence. And ask, when I compare with a suspect, what is the probability of a coincidence, of a coincidental match? Well, the probability of a coincidental match is the probability that the suspect matches the population. That is not looking at any evidence, you're just looking at a group of people. What's the chance the probability you found it by chance? And there, it turns out, and this is a concept that'll come back again and again, whatever the probability of that allele pair was in the genotype is the probability of its match with the suspect. So if you have a suspect with a definite allele pair, 10, 12, you can ignore all other values when you're computing a match. You just ask, in the evidence, what's the chance of a 10, 12? In the population, what's the chance of a 10, 12? This can become important. A standard question that I've been asked as, when I testify is, what if it was somebody else? Could it be some other allele pair? Well, yes, it could always be some other allele pair. It has some value, but it's not relevant, and that's the correct answer. When you're computing a likelihood ratio, which is the key statistic that is used in forensic DNA and much of the rest of forensics. In fact, it's the only statistic used in forensic DNA. And as we'll see, there are different ways you can use the data to get more or less information out of it. But it's the only DNA statistic. So what is it? Well, before I saw the data, the probability of a coincidence was whatever the match would have been in a population, like 5%. What's the chance that somebody in the room would have had it, would have had that particular allele pair genotype? Say 5%. Now I look at the data, I look at the evidence, and after I've looked at the evidence, there's a probability of some identification. Maybe it's very high. Maybe they exactly match. It's 100%. That ratio of the probability of identifying a suspect from the evidence divided by the probability of a coincidental match to the suspect, that ratio is the likelihood ratio. But you'll see that every statistic that we're talking about in this case, all three that were reported in court, are likelihood ratios, and they're simply pulling out more or less information. Okay, so you don't have to remember the word likelihood ratio, but the word information is really what's going on. At the suspect's genotype, to what extent is this an actual specific identification, or is this a random coincidence? And through the magic of probability, you just have to look at the probability of those genotypes and divide one number by the other, and the answer comes out. Okay, so the probability of identification is that the suspect matches the evidence. Of coincidence is that the suspect matches the population. And let's look at our example. If there's an exact match, it's 100%. If the 10-12 is what the evidence says, if 10-12 is what the suspect says, that's their genotypes, then the probability of a match is 100%. They're matching. You're looking at it. But now you have to divide by the probability of a coincidental match. What's the probability that the suspect matches the population? Well, how prevalent is that genotype in the population? We said it was 5% for a 10-12. And so you divide the 100%, the certain match that you have, for very easy data, by the 5%, the denominator that normalizes it and tells you how surprising this result is. And 100 over 5 is 20. And that would be, at that one locus of the 13, what the match score would be 20. And the reason you get large numbers in DNA is when you multiply 13 numbers like 10 or 20 together, you get a very big number with 12 or 15 zeros after it. Okay? So in real DNA evidence, crime labs and prosecutors or defense attorneys are not that fortunate anymore. You don't get as much perfect blood. In a property crime, the data are low level and mixed. In sexual assaults, the data is always mixed. In homicides, even, I guess criminals are just trying to get more work for the crime lab. Everything's mixed. So the new challenge for DNA for the last few years is not how to handle perfect evidence, where there's only one answer, like a 10-12, and it's obvious with just two peaks. It's what do you do with uncertain evidence? So here, like in our case, is a mixture of two individuals, say a victim and someone else. And then the lab goes through and does all of its work. It extracts the DNA, it amplifies it, millions of times over, and then it looks at what's there by seeing how much of each type of DNA is present, as it looks at the different sizes, the number of repeats. And now you don't have one or two alleles anymore, because you're no longer looking at one person. You're looking at something that's less certain. Okay, so one thing you can do is to throw up your hands and say, I don't know what to do with it, or you can attack it and say, oh, it's uncertain and DNA is supposed to be certain. But that's all silly, because the whole science of likelihood ratios and identification is designed to handle this, as I'll show you. So there's question data. Maybe we don't know which of these alleles are from who, but we do know that these two alleles, one or both of them had a 10, 11, and a 12, those three different length fragments. Okay? There are 4 alleles present, right? 2 from the mother and father of the first person, the mother and father of the second, so there are 4 altogether. One of them might be shared, and we're seeing some combinations. That's why we're seeing 3 alleles. Now when we make an inference, whether it's by computer or by a person using a very sophisticated method that might take 3 days of calculation, or a person using a very simple method that takes only an hour of calculation, if there is uncertainty in the 21st century, and even in the 18th century, uncertainty is represented with probability. So if you're going to quantify the extent to which you believe something is true, the probability, the odds with which this would happen, or something, the likelihood, then it's going to be different possibilities with associated probabilities. In this representative example, before we get into the actual case data, suppose that 10, 12, suppose we believe that the unknown was going to have a 12 in there regardless, but it could be a 10 and a 12 at 50% probability. It could be an 11 and a 12 at 30% probability, or a 12-12 at 20% probability. Different interpretation methods will give different percentages or different probabilities. I'm going to use percent, by the way, instead of writing out probability, because it means the same thing, and many people prefer to see big numbers like 50 as opposed to smaller numbers like 0.50 and all that. Okay, so it means the same thing. So now, if we have inferred this, notice there's evidence. A genotype was inferred from the data. There is no suspect involved. If you look at the suspect when you make an inference, you run the risk of bias. So you'd rather not do that, whether you're a computer or a person. So now that you've said what you believe the allele pairs would be in their probability, by whatever interpretation mechanism, you make a comparison. And suppose we're comparing at this one of 13 loci, this one location, the allele pair has the value 10, 12. What do you do? Well, we're going to do exactly what we did before. We're going to ask, what's the probability of a specific identification? Put that in the numerator. Ask, what's the probability of a coincidental match? Put that in the denominator. And it turns out that's exactly what the match statistic is going to be. Okay, so is it identification? Is it coincidence? We don't know, obviously, but we can tell you what the likelihood ratio is. To what extent is it one or the other? Before we saw the data, the probability of a coincidental match was 5%. Now afterwards, we're less certain. It's maybe 50%. It's no longer 100%, but that's okay. We'll form a likelihood ratio. So using those probabilities from the genotypes, before the probability the suspect matched the population, as you recall, we said would be 5%. Now that we've looked at the evidence, we may have more than one possibility, but at the suspect's genotype, all the evidence is saying is that we now believe, or a method believes, that it's 50%. It could be 10%, it's whatever you infer. It could even be less. That would be evidence in favor of the defense as opposed to the prosecution. It can go either way. In this example, the probability of an identification that the suspect matches the evidence, we said would be 50%. And now you see a decrease because 50 over 5 is 10, not the 20 that we had before. And so the take-home message of all this is that with proper DNA statistics and likelihood ratios, any method can handle uncertain evidence. What you do is you look at what the suspect's genotype is and ask, to what extent do you have a match from the evidence? Divide that by to what extent you have a match from the random population. That number, that ratio, is called a likelihood ratio. And then you multiply them together to combine all 13 loci to get the full match statistic. So that's all the math we're going to be doing. I left out all the big equations, but it's a ratio. I've heard people lecture on this. They'll give pictures of scales of justice and say, to what extent is it matching one way or the other. You can use all these metaphors and allusions, but basically, it's to what extent is it an identification? To what extent is it coincidence? And that's DNA match statistics. Okay, why don't I stop there?